Today we will be talking about history of microscope, types of optical and electron microscope. The invention of microscope dates back to the late 16th century, with the Dutch spectacle makers Hans and Zacharias Janssen credited for creating the first compound microscope around 1590. Later in the 17th century, Robert Hooke's micrographia introduced the microscopic world to the general public. Robert Hooke improved microscope design by using oil lamp lighting and Kotler's ball for better illumination. However, he encountered issues with image distortions, especially when using two lenses. In the late 16th century, Antony van Leeuwenhoek, a Dutch trapper and scientist, built a simple microscope with only one lens to examine blood, yeast, insects, and many other tiny objects. He was the first person to describe bacteria, and he invented new methods for grinding and polishing microscope lenses that allowed for curvatures, providing magnifications up to 270 diameters, the best available lenses at that time. In 1830, Joseph Jackson Lister discovered that using weak lenses together at various distances provided clear magnification. This was the prototype for the compound microscope. In 1872, Ernst Abbe developed a mathematical formula that allowed for the maximum resolution in the microscopes possible. And in 1903, Richard Sigmonti developed ultramicroscope that could study objects below the wavelength of light. In 1931, Ernst Ruska co-invented the electron microscope. An electron microscope depends on electrons rather than the light to view an object. Electron microscopes made it possible to view objects as small as the diameter of an atom. In 1932, Fritz Zernick invented the phase contrast microscope that allowed for the study of colorless and transparent biological materials. And in 1981, Gert Bennick and Heinrich Rohrer invented the scanning tunneling microscope that gives three-dimensional images of objects down to the atomic level. The powerful scanning tunneling microscope is the strongest microscope to date.
We will be talking about type of microscope. Basically, microscopes are of two types, optical and electron microscope. So these are the type of optical microscope and these are type of electron microscope. So to talk about optical microscope, it is divided into compound microscope and dissecting microscope. So these two are compound microscope and this is dissecting microscope.
Now I will talk about dissecting microscope. This is dissecting microscope and it is light illuminated. The image that appears is three-dimensional. We cannot see individual cells because it has low magnification. Some specimens are larger, so we do not have to magnify much. In this case, we can use dissecting microscope to get a better look at larger specimens. Now let's talk about the parts of dissecting microscope. This is eyepiece and it helps to magnify the image. The magnification of this eyepiece is 20x, so it will magnify the specimen 20 times. The next one is stage, where the specimen to be viewed is placed. The next is stage clip, which holds the specimen firmly. And this is adjusting knob. It helps to focus the specimen. This is illuminator and it has two types of mirror, plane mirror and concave mirror. It helps to illuminate the light to the specimen. The last one is base. It supports the whole microscope.
So talking about the compound microscope, these are the two types of compound microscope. But comparing this to this, we prefer this over this because this has only 40x magnification whereas this has 100x magnification. So we prefer this over this. So now I will be talking about parts of this compound microscope. Firstly, it consists of eyepiece. This is eyepiece. Now followed by eyepiece. So this is rotating nose piece. This rotating nose piece is connected with the objective lens. Now this is a stage. This stage consists of a stage clip and aperture. So below the stage, it consists of diaphragm. Below diaphragm lies the illuminator, and this is the knob that helps to adjust the aperture in the stage. And these two are the stage control. This is the arm, and this arm is connected with two adjusting knob. This one is coarse adjusting knob, and this small one is fine adjusting knob. So this is the controller which control the intensity of light in the illuminator. The final part of this compound microscope is base which support the whole microscope. So now I will be talking about different part of compound microscope and their functions. The first part of the compound microscope is eye peak. Eye peak is what you look through at the top of the microscope. Typically, standard eye peak have a magnification power of 10x. Optical eye peak of varying power are available, typically from 5x to 30x. The next part of the compound microscope is rotating nosepiece. The main function of rotating nosepiece is to hold the objectives. The objectives are exposed and are mounted on a rotating turret so that different objectives can be conveniently selected. And this is the stage. It is where the specimen to be viewed in place. A mechanical stage is used when working at the higher magnifications where delicate movement of the specimen slits are required. On the stage, we could see an aperture. Aperture is the hole in the stage where the light passes through to reach the specimen. Along with the aperture, there is also a stage clip in the stage. The main function of the stage clip is to keep the slits in place. Below stage is the diaphragm. The main function of diaphragm is to control the amount of light reaching the specimen. It is located right below the stage. This is fine adjusting knob. This knob is inside the coarse adjustment knob and is used to bring the specimen into the sharp focus and make the view of the image clear and better. The part that you can see is illuminator or light source. It is the light source of a microscope, typically located in the base of the microscope. The last part of the microscope is base. It is used to support the microscope.
Hello everyone, in this video lesson I'm going to talk about the types of electron microscope and some uses. Before we move on to the types of electron microscope, we have to know the definition of the electron microscope. Electron microscope is a device which uses a beam of electron to illuminate and magnify the object at the high resolution. And now let's talk about the types of electron microscope. Basically, there are three types of electron microscope: personally, scanning electron microscope, transmission electron microscope, and confocal electron microscope. And now let's talk about the first type of electron microscope, that is scanning electron microscope. And you can see in the above picture, this one is the scanning electron microscope. And scanning electron microscope is a type of electron microscope that produces the image of a sample by scanning it with a focal beam of electron. And scanning electron microscopes are widely used in various fields including material science, biology, and nanotechnology, and also used to observe the surface topography and composition of the sample with a high resolution. And now let's move on to the second type of electron microscope, that is transmission electron microscope. Transmission electron microscope is the another type of electron microscope that transmits a beam of electron through the sample to create an image. And the specimen the use for the transmission electron microscope it should be 20 to 100 nanometer thick. Moreover, this kind of electron microscope are usually used to study the internal structure of a specimen.
Confocal electron microscope is a specialized type of electron microscope that uses confocal imaging technique to produce three-dimensional images of a sample with high resolution. It combines the principles of confocal microscopy, which is commonly used in light microscopy, with electron microscopy. Confocal electron microscopes are particularly useful for studying the three-dimensional structures of biological samples and materials at the nanoscale. They provide detailed insights into the spatial arrangement and distribution of features within a sample.